And today what we're going to focus on is types of inheritance that don't follow Mendel's rules. Keep in mind, Mendel did his work roughly 160 years ago. So at the time, people didn't know about meiosis. They didn't know DNA was a genetic material. So not too surprising that Mendel got some things right, but didn't really describe the way that all of inheritance works. In fact, if you remember, I said he started out studying 14 characteristics, and at the end, he only wrote about 8 of them, or 7, I'm sorry. That was because the other 7 didn't really follow his rules, his laws that he devised. And it turns out the other 7, still typify the way inheritance occurs. Mendel's laws turn out to be really just the basics of how characteristics are inherited. So let's look at, first of all, remember that Mendel's first law was his law of particulate inheritance. He said that, inheritance is controlled by 2 factors, particles. Those would be the things that we think of as homologous chromosomes today, or the alleles that you get for one gene. And Mendel said that one of these factors is dominant, and the one that's dominant can hide the recessive factor so that we can't see it. He used this to explain how characteristics had disappeared when he crossed his true breeding lines. So let's look first of all at blood types, because blood types really violate Mendel's laws in 2 different ways. First of all, if we look at what's called the ABO blood type, there are 4 different blood types within this system. You could have type O blood, you could have type A blood, you could have type B blood, or you could have AB. Now, there are actually 3 different alleles here. If we would look at you or we would look at me, an individual person still only has 2 alleles. But if we would look at the entire population, there are 3 different alleles out there. There's an I with a big A. And the I with the big A produces they're called antigens. Antigen is just the fancy name for proteins that the immune system can recognize. So there's an I with an A. And notice that this is written the same way as X-linked characteristics were the other day, things that were on the X chromosome, where we had a letter up above the line, a superscript. And we said that X and whatever the letter was, we were looking at big B and little b, that was a unit and it couldn't be separated. So this I and this A are a unit that can't be separated. When we're working a problem with this, they have to stay together throughout the entire problem. If you inherit a capital I with a big B, this is what causes type B blood, because you get the B antigens. And when we talk about the A antigens and the B antigens, you might have heard about antibodies in the past year. There was talk of developing an antibody test for COVID-19. And antibodies are protein that your body makes that react to antigens. And your body makes antibodies that react to antigens that you don't have. So somebody with type A blood makes antibodies against the B protein. Somebody with type B blood makes antibodies against the A protein. But you could inherit an I capital A from your father and an I capital B from your mother. And if you do, you have type AB blood. And if we would look at your red blood cells, you have some of the A antigens and you also have some of the B antigens. You have both on your blood cells. Now notice, if you have type AB blood, you don't make antibodies that attack the A antigen because you would end up attacking your own blood. And you don't make antibodies that attack the B antigen because, again, you would end up attacking your own blood. So two of the three alleles are the I big A and the I big B. The third allele is little I. And little I can cause type O blood. In little I, you don't make A antigens or you don't make B antigens. So notice that with little I, to have type O blood, you have to be little I for your genotype. Because if you get one I big A or one I big B, you won't have type O blood. You'll have, whatever you inherit, that other allele. So in this case, there are three different possible alleles for someone, for their blood type. Now, as I say, you still only have two. You got one from your mother, you got one from your father, but if we look at a group of people, we would find all three. Now, notice this is also a good example. Even though little I is recessive, if we go most places around the world, type O blood is one of the most common blood types. Most places around the world, if we look, type O blood is about 45% of the population. And type A blood is also about 45% of the population. And B and AB are both about 5% of the population. So they're relatively rare. So there are three different types of alleles here. So that violates Mendel's idea that there's one dominant factor and one recessive factor. Now, another way that this violates Mendel's original ideas is, notice that I big A is dominant over little I. If you have type A blood, for a person with type A blood, you could be I big A, or you could be I big A, little I. So type A is dominant over type O. If you're I big B or I big B, or I big B, little I, you have type B blood. So the allele for type B blood is also dominant over little I. So type A and type B are both dominant. Well, Mendel talked about one dominant factor and one recessive factor. And when we look at these two factors for somebody with type A blood, as I mentioned before, they have some of the A antigens in their red blood cells. They have some of the B antigens in their red blood cells. So they have both. So if you think about it, remember the phenotype is the physical appearance. If you look at somebody with type AB blood, you can see the phenotype that we see in somebody with type A blood, because the phenotype is really these A antigens. And you can also see the phenotype that we see with someone with type B blood. In this case, the phenotype is these B antigens. So when we can see both of the phenotypes, so somebody who's blood type AB is I big A, I big B. So if we look at them, we can see both phenotypes from both alleles. This is called codominance, when you can see both of the phenotypes. So notice this violates Mendel's idea about dominance. First of all, there's two alleles that are dominant. Secondly, if they're together, one of them isn't dominant over the other. If your genotype is I big A, I big B, we can see the A protein and we can see the B protein when we look at your red blood cells. So one of those isn't dominant and one of those isn't recessive. Usually when we're reading about something that shows codominance, the clue that we'll get is that they'll talk about individuals that are heterozygous, because individuals that are heterozygous have a different phenotype. So notice for the blood types here, there are 4 phenotypes: type O blood, type A, type B, and type AB. And these reflect the types of proteins, the type of antigens that we see in your red blood cells. Now, keep in mind, there are lots of antigens in the red blood cells. There's another type of antigen called the Rh antigen. And if you have the Rh antigen, that's when you're A positive or B positive or O positive. If you don't have the Rh antigen, then you're A negative or B negative or O negative or AB negative. So there are lots of different antigens. So when we say somebody with type O blood doesn't have these antigens, we're only talking about the A antigen and the B antigen. We're not talking about every antigen that's found on blood cells, just the ones in this category. So we want to keep that in mind. Now, there's another type of inheritance that violates Mendel's law called incomplete dominance. And as you can tell by the name, this doesn't follow Mendel's law of dominance. Now, if we think about codominance that we just talked about, and we think about incomplete dominance, the basic difference that we're going to see is what we observe when we look at the phenotype of heterozygous individuals. So remember, the phenotype is their physical appearance. So in incomplete dominance, when we look at heterozygous organisms, we don't see both phenotypes. So it's not like type AB blood where we can see both proteins. Basically, what we see is something that's kind of in between the two homozygous individuals. So there are a lot of flowers where plants that are big R make red flowers, and plants that have a genotype of little r make white flowers. But plants that are heterozygous, big R, little r, don't make red flowers with white spots or white flowers with red spots. We can't see both colors. We get pink flowers. We get something kind of in between the two, like we would see if we were mixing pigments in a paint set. So really, codominance and incomplete dominance are very similar in that sense. The big difference between codominance and incomplete dominance is that when we look at codominance, we see the phenotypes of both homozygous individuals. We see the A proteins, we see the B proteins. In incomplete dominance, when we look at someone that's heterozygous, we don't see red and white. We see something in between the two. Like I say, think of if you mix two paints together, red paint and white paint, you get pink, you get a lighter version of red. Now, again, the key is when we're looking at inheritance, if something is inherited through incomplete dominance, then the problem is going to tell us something different about heterozygous individuals. Normally, if we have complete dominance, we know that heterozygous individuals show the dominant phenotype. But if we have codominance or incomplete dominance, that's not true. So we have to be told what it is. So there are some guinea pigs that if they're big Y, they have yellow hair. If they're little y, they have white hair. And if they're big Y, they have what's called a cream colored hair, which you could think of as being kind of a light yellow or kind of a dirty white. When we think about incomplete dominance, heterozygous individuals have a phenotype that's kind of partway in between the two homozygous possibilities. Now, keep in mind with both of these, one of the things we do that, if you think about our usual rules, normally the capital letter indicates the dominant allele, and the lowercase letter indicates the recessive allele. And notice for incomplete dominance, we still use big R, usually to designate red, little r to designate white. But if you think about it, big R isn't really dominant over little r. Because if red was dominant over white, then if we looked at a plant that was heterozygous, it wouldn't be pink, it would be red. So, we have to keep in mind that even though normally capital letters are dominant alleles, that rule isn't always followed. Now, another type of inheritance that doesn't really follow Mendel's laws is something called polygenic inheritance, or it's sometimes referred to as quantitative inheritance. And we think that this is probably the most common type of inheritance in people and in other organisms as well. We looked at characteristics the other day when we were looking at our own genetic characteristics. We looked at characteristics that are determined by one gene. You know, one gene determines whether you have widow's peak or a straight hairline. One gene determines whether you have a bent little finger or a straight little finger. So in polygenic inheritance, remember earlier in the semester, we talked about polypeptides and polysaccharides. Polygenic inheritance, many genes affect one particular aspect of the phenotype. A lot of times these types of characteristics, this is also referred to as quantitative inheritance. And this is because when we look at these characteristics, there's usually a whole spectrum or range of possibilities. So think about human height. Height is a good example of a polygenic inheritance. You know, people aren't just 5 feet tall exactly, or 6 feet tall exactly, or 7 feet tall exactly. People are 5'1", or 5'2", or 5'3", or 5'4", or 5'5". They can, and we can go into more precision than that. People aren't necessarily 5'5" or 5'6". People are 5'5" and a half, or 5'5" and 3/4, or, you know, depends on how precise our ruler is, how well we can measure this. So when we have polygenic inheritance, we have a lot of genes. Now, typically a lot means more than one, but we have multiple genes that affect the inheritance of one part of the phenotype. So as I say, height is a good example of a polygenic characteristic. You know, there are lots of heights, and typically when we have something that's polygenic, also, you know, one height blends into another. As I say, it's not you're either 5'5" or 5'6". You could be anywhere in between 5'5" and 5'6". Skin color is another polygenic inheritance. There are lots of skin colors that kind of blend into one another. You know, there's not just a few possibilities with nothing in between. Intelligence, although we don't have a very good definition of what intelligence is polygenic. There are a lot of different genes that affect intelligence. So most of the studies that have been done recently show that pretty much all phenotypic characteristics in people are polygenic or controlled by quantitative inheritance. Even a lot of the ones that we used to think only involved one gene. Turns out when we study them in more detail, they do involve multiple genes. Now the problem is when we look at polygenic inheritance, trying to make Punnett squares like those Punnett squares that we did the other day, that becomes very complex. So we did some monohybrid crosses the other day, we did some dihybrid crosses, and we're not going to start looking at polygenic inheritance because that gets a little more complicated than we want to go into. Now there's one particular type of polygenic inheritance that we do want to talk about a little bit, and this is something called epistasis. And in epistasis, the alleles at one gene affect the phenotype that you get at another gene. And a good example of this is Labrador retrievers. So let's look at Labrador retrievers. And with Labrador retrievers, there are two primary genes involved in their color. One is big B and little b. And they both affect the amount and the type of melanin that Labrador retrievers make. And when we look at these, big B produces a darker color, little b produces a lighter color. And this really affects the color of the skin primarily, but it can also affect the color of the eyes. There's another gene called the extension gene, which we indicate with E's. And capital E means the dark color doesn't only appear in the skin, it also appears in the hair. The recessive allele at this spot, little e, means that the dark color is limited to the skin. It doesn't appear in the hair. So let's look at the three types of Labrador retrievers. So for a Labrador retriever, if it has one capital E, could have two, but it's got to have at least one. If it has at least one capital E and one capital B, now the capital B means it will make a darker variety of melanin, and the capital E means that melanin will appear in the skin and also in the hair. And we get something called a black retriever. If we have at least one capital E, but then we have two lowercase b's, so we don't have any capital B's, the color appears in the hair, but it's not the darker version of melanin, it's the lighter version. So we get what are sometimes called chocolate labs. And if we have two little e's, this means that the dark color, the melanin, doesn't appear in the hair. If you kind of, run your fingers through their hair and you look at their skin, they can still have a dark skin, but the hair that covers the skin is lighter. And these are golden labs or yellow labs a lot of times. Notice they're not the same as golden retrievers, even though Labradors are sometimes called Labrador retrievers. This is different than a golden retriever. So there's three different colors, depending on whether they get the allele for dark melanin and they get the allele that color goes into the hair. They get the alleles for light melanin and they get the allele that goes into the hair. Notice if they get the two alleles so that the color is confined to the skin, doesn't matter what they have for the two B's. They're going to look like this. They're going to be a yellow retriever or a yellow lab. So we want to keep that in mind. Now, people have the same thing. We were talking about skin color for a moment ago. If we look at skin color, there are at least 4 different genes that affect skin color in people, probably more like 16, but once you get more than about 4 or 5, our technology isn't very good at figuring out exactly how many genes. But there's also a gene that if you inherit one of these genes, you could be an albino. And then you're kind of have a skin color that's very different. If we think at albinos, if you get homozygous recessive at one of 7 different genes, this can prevent your body from making something called eumelanin or pheomelanin, which are 2 varieties of melanin that give your skin its color. And in that case, you'll be an albino. So when you are homozygous recessive at one of these 7 genes, it doesn't matter what you have at the other 6, you're going to be an albino. Now, there are different types of albinos, but that still will end up being an albino of some sort. Now, the reason I say that there are different types of albinos, because these alleles that cause albinism are a good example of something called pleiotropy. And at least when we think about it, pleiotropy is basically the opposite of polygenic inheritance. In polygenic inheritance, we have many alleles that affect one aspect of your phenotype. In pleiotropy, we have one gene, one pair of alleles, and they affect several different parts of the phenotype. So an example is what we just talked about. When we look at albinos and when we look at people who aren't albinos, it turns out that this doesn't only affect skin pigmentation, it also affects your eye and your hair pigmentation. If you've ever seen some people who are albinos, there's a guitarist named Johnny Winter who's not very active anymore. He's older, but he was an albino. And if you ever would have seen him in concert, his eyes looked like they were pink. He didn't really have pink eyes in the sense that you might have brown eyes or blue eyes or green eyes. Basically, the iris of his eye, the part where there's some color normally, was clear. And the pink that you were seeing was actually the light coming into his eyes and reflecting off the blood vessels in the back of his eyes. And in fact, one of the problems that albinos who have this condition have to be careful about is they're much more susceptible to basically burning their retina, the back of the eye, which is where the cells are that detect light. So they're much more susceptible ultimately to going blind and to getting cataracts and things like that. So a lot of times in normal situations when they're indoors or whatever, people who are albinos have to wear sunglasses because their eyes are very sensitive to light, because one of the things that the brown and the green and the different colors in our eyes do is they screen out some of the light so that it's not too bright, so that it doesn't overwhelm the cells in our retina. Another example of pleiotropy is something called polydactylism. Poly again means many, and your dactyls are your fingers, are your toes. So in polydactylism, people have extra fingers and extra toes. So a few years ago, there was a pitcher for the Chicago Cubs named Al Fonseca, and he had a sixth finger. Now, unfortunately, the sixth finger doesn't have a very good nerve supply, and it doesn't have very strong musculature. So the sixth finger is kind of like an even wimpier vision of your pinky. So it's not very useful. So even though Al Fonseca had six fingers, he couldn't throw like really wicked curveballs or anything. He was a pretty so-so pitcher. He wasn't all that great. Yeah, you can't play like really cool chords on a guitar or anything. Polydactylism is another example because it can affect your fingers or your toes. So in one of those weird coincidences, one semester, when we were here on campus, we could actually, we looked at genetic characteristics one semester in class. I actually had two girls who both had six fingers. They weren't related or anything, but just very unusual because that's a very rare characteristic. So having two people the same semester who had that was extremely unusual. Now, the other thing we have to keep in mind is, for the last couple of days, we've been focusing on really how biology affects you. What do you inherit from your parents? But we have to keep in mind there are also environmental effects on the genes. One of the things we know is that while a fetus is in the mother's womb, nutrition and also nutrition the first few years have a big impact on your adult height. If you're malnourished while you're in the mother's womb, especially if you don't get enough protein, and if you don't get enough protein the first few years after you're born, you'll never reach your genetic potential for your adult height. Because remember, proteins are used by the cells to do everything, and they're used to build a lot of parts of the cell. When we think of bone as being mostly calcium, but actually bone is mostly a protein called collagen. So if you don't get enough protein while you're a fetus or while you're a small baby, you won't reach your utmost potential for height. And that's true even if you get more protein later in life, because this is something that's time sensitive. Now, if you think about it, remember that we talked about height a moment ago in connection with something that is polygenic. And most polygenic characteristics are strongly influenced by the environment. A lot of you have probably noticed in the last month or so, if you go out, if you're in the sun regularly, there can be a change in skin color. You might tan or burn or ash. There's all sorts of different possibilities, but this is because when you're out in the sun, your body starts making more melanin, because the melanin is a protective chemical, is one of the main things it does. Stops sunlight from penetrating too deeply into your skin, because when sunlight penetrates deeply into your skin, the ultraviolet wavelengths can actually cause skin cancer. And intelligence is like height. If you're malnourished, especially if you don't get enough protein, your intelligence never develops to its full potential. And again, this is more important for the nutrition you get when you're a fetus or when you're a young baby. Once you're 40, if you eat extra protein, it won't make you smarter, sorry. So now we want to talk about one more type of inheritance that violates Mendel's laws. And this really violates the law that Mendel came up with when he was doing his dihybrid crosses. And remember, I've mentioned several times, he started out studying 14 characteristics. He ended up only studying 7. Well, it turns out that when we think about Mendel's law that he ended up formulating after his dihybrid crosses, he said that the inheritance of one characteristic doesn't affect the inheritance of other characteristics, something that was called his law of independent assortment. Well, it turns out that the 7 characteristics that Mendel threw out that he decided not to study, that wasn't true for them. They were inherited, but whether they were inherited or not depended partly on what you inherited from other genes. And it turns out this is true for anything that we call a linked gene. Keep in mind that when you make an egg or a sperm, what meiosis does is it separates the homologous chromosomes. If I make a sperm, some of my sperm get chromosome number 1 that I inherited from my mother. Some of my sperm, about 50%, get chromosome number 1 that I inherited from my father. Well, the average chromosome in a person has about 1,000 genes on it. And since you inherit a whole chromosome, you tend to inherit the genes that are on the chromosome together. Now, you don't always, because remember, one of the things we talked about during meiosis was crossing over, where a little piece of chromosome 1 from your mother gets put onto chromosome 1 from your father. And the corresponding piece of chromosome 1 from your father goes on to chromosome number 1 from your mother. So crossing over can separate genes that start out on the same chromosome, but mostly genes that are on the same chromosome are inherited together. So they don't follow Mendel's rule or law of independent assortment. So when they're on the same chromosome, we say that they're linked, that you usually inherit them together. So the only place where we don't really have crossing over occurring, if we look at the human chromosomes, is the X and Y chromosome. Because unlike the other chromosomes, if we look at chromosome number 1, the genes on chromosome number 1 are the same, whether you inherit that from your mother or your father. Now, remember what we said earlier. This doesn't mean the alleles are the same. This doesn't mean they have the same instructions. You can inherit the allele for blue eyes from your mother, brown eyes from your father. But the instructions, the gene for eye color is at the same location on the same chromosome in both cases. But the X and the Y are different. The X and the Y don't have the same genes on them. So we don't generally have crossing over between them. Now, we know that males are XY and females are XX, and notice this is true for all mammals. It's not true for all animals. If we look at birds, in birds, females have 2 different chromosomes, and the two chromosomes in males are the same. And if we look at a lot of plants don't even have male and females. Many plants make male flowers and female flowers on the same plant. So a lot of plants don't have sex chromosomes at all. So we want to keep that in mind. Now, if we think about the difference, men have to get their X chromosome from their mother because they have to inherit the Y from their father. So notice one of the things this means is if we look at a male and he has a disease that's X-linked, which means it's on the X chromosome. If we have a disease that's X-linked and my dad has it, I won't inherit it because it will be on my father's X chromosome and I'm going to inherit his Y chromosome. Now, on the other hand, for women get one X chromosome from mom and one X chromosome from dad. So women can potentially inherit an X-linked disease from their father. Men can only inherit an X-linked disease from their mother. And notice usually when we talk about the X and Y chromosomes, we focus more on diseases that are carried on the X chromosome because males get one X, females get two X, so it does affect both genders. We don't really focus too much on the Y chromosome for one simple reason. Only males get the Y chromosome, and males only have one Y chromosome. So if I have a genetic disease on my Y chromosome and I have a big family, if I have 10 boys and 10 girls, if I have a genetic disease and the gene is on the Y chromosome, none of the 10 girls will inherit it. All of the 10 boys will inherit it. So usually the pattern of inheritance for something that's Y-linked is very obvious. A male has it, his sons get it, his grandsons get it. None of the girls get it. The pattern is not always as obvious when we're talking about something that's X-linked. Now, if we think about what it means for alleles to be dominant and recessive, a dominant allele always shows up in your phenotype. So females have two X chromosomes. So this means they're slightly more likely to get genetic diseases that are X-linked and dominant. You could think of it like buying a lottery ticket. Unfortunately, in this case, the lottery is you get a genetic disease. That's what the winner gets. But a female is buying 2 tickets for the lottery. Males only have one X chromosome, so they're only buying 1 ticket for the lottery. So females are slightly more likely to get genetic diseases that are X-linked and dominant. Notice it's not a big difference. For most diseases that are X-linked and dominant, what we find is that, maybe 52% of the people who have it are female and 48% are male, or 55% are female. However, if we look at genetic diseases that are X-linked and recessive, men are much more likely to get genetic diseases that are X-linked and recessive. And the basic reason why is if we look at genes that give you diseases, they tend to be relatively rare. So most of the diseases, whether they're dominant or recessive, are pretty rare. Most of the genes that would cause the normal condition are much more common. So what this means is, if you think about it, so if you look at a male has one X and one Y. So if we look at something like red-green color blindness, which is an X-linked genetic disease, if a male gets one allele for red-green color blindness, and he would inherit that from his mom, he's going to have the disease because he doesn't get another allele. He gets the Y from his father. But if you think about a girl who gets an X chromosome with the allele for red-green color blindness still gets another X chromosome from the other parent. And if you think about what I just said, the alleles for the normal condition are more common. So the other X chromosome she gets probably has an allele for normal color vision. So men are more likely to get genetic diseases that are X-linked and recessive. So X-linked and dominant occur more frequently in females, but it's kind of close. Maybe 55% of the time in females, 45% in males. Maybe 52% in females, 48% in males. It's not the same for every disease. But if we look at things that are X-linked and recessive, not only do they occur more frequently in males, much, much, much more frequently in males. If we look at red-green color blindness, about 92% of the people who have red-green color blindness are men. Only about 8% are women. So it's relatively rare for women to get these. You know, we had that color blindness test we looked at the other day. Usually when we're on campus, you know, I typically have 2 or 3 classes. And out of the 2 or 3 classes, I almost always have one male who's colorblind. And I think I've been teaching this class for about 20 years or so. All the years I've taught this class, I've had like 2 women in all the 20 years. And I have 1 or 2 men every semester. So that tells you how much more common it is in men than in women. And finally, another thing to keep in mind is there's another kind of genetic disease that you can get when the homologous chromosomes or sister chromatids don't separate during meiosis, when you're making the eggs or the sperm. Their separation is something called disjunction. So non-disjunction means they don't separate. So what happens if they don't separate? You're going to get some eggs or sperm that have either extra chromosomes or not enough chromosomes. So if we look, there's really only 2 situations normally where you can get extra chromosomes. Trisomy 21, which is Down syndrome, is probably the best known example of non-disjunction. Not always, but usually it happens because of a problem with meiosis in the mother's egg cell. And the baby inherits 2 copies of chromosome 21 from mom and 1 copy from dad, but can be the reverse. It's just more common coming from the mother. But the same thing can happen. You can inherit extra X's or extra Y's and survive. So some people, and notice they would be males, are XXY, and this produces something called Klinefelter syndrome. The symptoms aren't always similar, except males with this syndrome are almost always sterile. Now, how do we know this is a male? It's got 2 X's like a female, and it's got a Y. If you have a Y, you're going to be a male. Super males have the genotype XYY. A lot of times there are different types of learning disabilities, but this is very common. It turns out that about 1 in every 1,000 males actually has an extra Y. So if you think about the population of the United States, you know, 300 million people, a little bit over, you know, that means that basically there's about 300,000 people with super male syndrome. XO, which indicates a female who receives 1 X chromosome and then doesn't get a second chromosome. Females with this genetic condition have what's called Turner syndrome. And again, they're often sterile. And this is the only case of what's called monosomy, getting 1 copy of a pair of homologous chromosomes, that is known to be survivable on a regular basis. There are a few cases that have been documented of somebody getting only 1 copy of chromosome 13 or 1 copy of chromosome 18 and surviving. But most fetuses that get 1 copy of chromosome 13 or 1 copy of chromosome 18 don't survive to be born alive. So if you get extra copies, or if you don't get enough copies of the chromosomes, this is usually fatal with a few minor exceptions. And notice if you think about some of the exceptions, you know, trisomy 21, remember what we said, chromosome pair 21 is actually the smallest pair of chromosomes. So there seems to be some problem with getting extra copies of a lot of genes. Since chromosome 21 is relatively small, it doesn't have that many genes on it. And notice if we look, remember the chromosomes are numbered and the smaller numbers are bigger. Notice that the other cases where you might survive, chromosome 13 or 18, they're not in the top half. They're not real big chromosomes. So, you know, that may be a reason why occasionally people survive with that. So these are all conditions that violate Mendel's laws, but remember what we said. Mendel did his work about 160 years ago. People didn't know about meiosis. They didn't know about DNA being the genetic material. So from a practical standpoint, it's not very surprising that some of what Mendel, you know, thought were laws that governed all of inheritance turn out to be things that there are a lot of exceptions to. So are there any questions about non-Mendelian genetics before we leave this topic? Okay, we're going to switch screens and we're going to look at some problems with non-Mendelian genetics. So let's look at a problem with incomplete dominance, and notice codominance works the same way. Now, remember what I said the other day, when we work genetics problems, the problems will usually start out and give us some information about which allele is dominant, what type of phenotype it causes, which allele is recessive, what type of phenotype it causes. If the problem has to do with incomplete dominance or codominance, the key is it's going to give us some information about 3 different phenotypes. What happens if you're homozygous? What happens if you're heterozygous? And what happens if you're, well, I should have said homozygous dominant, homozygous recessive, and heterozygous. So let's look at this one. In snapdragons, red flowers are caused by an allele big R. White flowers are caused by an allele little r. The heterozygote, big R, little r, has pink flowers. So this is going to be our hint when it tells us that the heterozygote has a certain phenotype and it's different than the parents. Now, depending on how you like to do problems, I always think that, especially with these, but sometimes with genetic problems in general, that I sometimes think it's a good idea to kind of write down what you're given at the beginning. So this is kind of the information that we should get from what we're given at the beginning of the problem. So what genotypes are possible for a white flower? Well, if we're talking about a white flower, the only genotype that would give us white is little r, little r. What's the phenotype of a plant whose genotype is big R, little r? Well, that plant is going to be pink. What do you predict for the genotypes and phenotypes of the offspring from a cross between a red flower and a white flower? Okay, so let's work this out. I think you'll see that we probably don't have to do a Punnett square here, but we will to show our work. Keep in mind when we have the test over this material, you will have to do a Punnett square. Well, doesn't really tell us, so we'll assume that the male plant is big R, the red plant, and the female is little r, the one that makes the white flowers. So if we look at the male, we were looking at an animal, we would call it sperm, but it's really pollen on a plant. All the pollen that it makes are big R. all the eggs that it makes are little r. Now remember what we said about our Punnett square. What we're going to do is we're going to basically make a square, and we're going to put all the different types of eggs that the female can make across here. We're going to put all the different sperm, or pollen in this case, that the male can make over here. So remember, the way we fill out our Punnett square, we carry whatever the male can make across to every cell in the same row. We're going to carry whatever the female can make down to every cell underneath it in the same column. Well, we probably didn't have to do a Punnett square realistically on this one, because the male can only make one type of pollen, the female can only make one type of egg, so they can only make one type of offspring. So what do you predict for their genotypes and phenotypes? Well, you can never leave your answer in the Punnett square. All of them are big R, little r. That's their, what, genotypes. What about their phenotypes? Well, their phenotypes, they all make pink flowers. Okay, now let's look at D. What do you expect for the genotypes and phenotypes from a cross of two pink flowers? Okay, if dad produces pink flowers, he's big R, little r. If mom produces pink flowers, she's big R, little r. Now, what type of pollen can dad make? What type of gametes? So notice, to list what type of gametes he can make, we have to separate our R's. So I'm going to put a comma in between them. If we just write big R, little r again, we didn't write the gametes, we wrote the phenotype. Because remember, the gametes only carry one of the alleles. The other allele comes from the other parent. Now, since mom has the same genotype, mom can make the same two types of gametes, big R, little r. So let's make a Punnett square here. Okay, so we've got big R in one of the cells. We've got little r in the others. So remember, we're going to carry the big R down to each cell underneath it. We're going to carry the little r down to each cell underneath it. Now, the father can make two types of pollen. So we're going to put each type of pollen as the kind of header for one row. And then we're going to carry that across to everything else in the same row. So if we look at this, I can spell, for the genotypes, notice we have a total of 4 cells in our Punnett square. So if we look at these 4 cells, a fourth of them are big R, little r. Sorry, big R, big R. 2/4 of them are big R, little r. One here and one here. And then finally, a fourth are little r, little r. And remember what we said, these should add up to 4/4, because If they add up to 4/4, then we counted everything once. So if you think about this fraction, the denominator, the 4, comes from the fact that there's 4 different cells in our spreadsheet here. The 1 and the 2 and the 1 come from how many cells have that particular type of, that particular genotype. Okay, then if we look at the phenotypes. Okay, the 1/4 that are big R are red. The 2/4 that are big R are pink. And the 1/4 that are little r are white. Oops, I guess I should write flowers in here. Now notice, instead of fractions, if you prefer, you could write decimals. Or if you've had math and you know what frequencies are, you could write frequencies, which would be 0.25 and 0.50 and 0.25, basically a decimal representation. So now let's think about a sample problem with X-linked inheritance. Now, when we read about a problem with X-linked inheritance, here's the key. Somewhere in the problem, it's going to tell us it's X-linked. And this is really the only time that we care about whether individuals are males or females. Because for genetic characteristics that aren't X-linked, that are on one of the first 22 pairs of chromosomes, what a male inherits is exactly the same thing as what a female inherits. It's only when we start talking about the X or the Y chromosome and the genes on it that males are different from females. So the dominant allele produces normal blood clotting. Okay, so the dominant allele here is going to be X big H. I think I'm going to need to make that font size bigger. Hemophilia is sometimes called bleeder's disease because people who have this, their blood doesn't clot normally. Now we have the protein, of course, is involved in this. That's why we have instructions on the DNA. Remember, the DNA is instructions for making proteins. If we look at people who lack this protein, their blood doesn't clot, so they bleed to death very easily. So now we have a protein that we can make synthetically, and we can give you this protein and your blood will clot. But if you go back more than maybe 60 or 70 years, people who had hemophilia always died when they were a very young age. Because especially if you go back in time, maybe 150 years or so, when, you know, a lot of people grew up in rural environments and you had to work on a farm, you know, you work out in the fields, you get a little cut on your hand. Except if you have hemophilia, you bleed to death from a little cut on your hand. And if you go back 150 years, your father could potentially have hemophilia. Probably have to come from a wealthy family so that, you know, he was kind of never had to do physical labor. But your mother would never have hemophilia. Because for a woman, if she has hemophilia, she would bleed to death when she reached puberty. So sometimes we have genetic diseases that affect the two genders differently. So what's the genotype of a man who's a hemophiliac? So a man who's a hemophiliac, now he only gets one X chromosome, and that X chromosome has to have a little h on it. His other sex chromosome is a Y, and because this is an X-linked characteristic, it's on the X chromosome. If it was Y-linked, then it would be on the Y chromosome, but not on the X. What's the genotype of a man with normal blood clotting? Well, a man with normal blood clotting is X big H, Y. So the fact that the male only gets one X, this is why men are more likely to get hemophilia, for instance. Because if a male inherits a little h, he doesn't get another X chromosome with another H. If a woman inherits an X with a little h from one parent, she probably gets an X with a big H from the other parent, because the allele for the normal condition for not having the disease is generally more common. So what's the genotype of a woman with normal clotting blood if her father was a hemophiliac? So a lot of times when you're asked questions like this, one of the best ways to try to solve them is to go through it kind of step by step. Okay, well, I guess we should start up here. Okay, she's a woman, so we know we're going to have two X chromosomes. She's got normal clotting blood. Well, if she's got normal clotting blood, oops, then she's got to have an X with a big H. But her father was a hemophiliac. So this is her father. Well, if this is her father, notice she's going to get the X with the little h. She doesn't get the Y, because if she did, she wouldn't be a she, she'd be a he. So she's going to be X big H, X little h. So she will not have hemophilia. Notice this problem doesn't say anything about incomplete dominance or codominance. So this means it's normal dominance. If you get one big H, like this woman did, you will have normal clotting blood. So we would not be able to look at this woman and see that she was a hemophiliac. Now, realistically, if this was that woman's father, we would probably know that he was a hemophiliac. Or if she had boys, if you think about this, when she becomes a mother, if she has boys, it's a 50-50 chance they get the X with the big H or the X with the little h. So if she has a large family, yeah, it's quite possible that she would have some boys who have hemophilia. So this really caused a big problem back when AIDS was first discovered. Because AIDS is a sexually transmitted disease. And when AIDS was first discovered, a lot of the people who were diagnosed were young boys, and they had AIDS. Well, why did they have AIDS? Because even if they weren't gay or anything, if you're a hemophiliac, a lot of times you have to receive regular blood transfusions. Anytime you get a minor cut or scratch, you lose blood. So you have to get a transfusion to replace that. And when we first discovered AIDS, we couldn't screen the blood supply and eliminate tainted blood that had the HIV, the human immunodeficiency virus, in it that causes AIDS. So there were a lot of famous instances. There was a child in Indiana who became an AIDS activist because he got AIDS through a blood transfusion. Unfortunately, he ultimately died. So let's look at D, a man with normal clotting blood. Okay, so a man with normal clotting blood. And since we're going to work a problem, I'm going to color code things again so I can keep track of mom and dad. So, okay, a man with normal clotting blood marries a heterozygous woman, okay? So she's heterozygous. So she's big H, little h. Basically, she's like this woman we talked about up above. Now, what type of sperm can the male make? Well, if we look at this male, for the male, he can make two types of sperm. Half of his sperm will carry big H. Remember, your sperm are always made in equal numbers, whatever the options are. Half of his sperm will carry the Y. Now, what type of eggs can the female make? Well, for our female, half of her eggs carry the capital H. And half of our eggs carry the little h. Now notice you might be tempted to say that, wouldn't her body make more of the eggs that have the big H? No. It's a random process. So even though one of these chromosomes has the gene for a serious disease on it, they're both made in equal amounts. Okay, so now we're going to make our Punnett square to figure out what type of results we see for our children. Now, one of the crucial things we have to do in an X-linked problem is we have to remember that when we're doing our X-linked problem, we have to keep the X and the H together. They're a single unit, so they can't be separated. So we're going to do the same thing we always do. We're going to take however many different eggs that the female can make, and we're going to write those down across the top, and then we're going to carry them down into each cell underneath. Then we're going to take the sperm that the male can make, and we're going to carry those across to each cell in the same row. Now, up till now, this has been, fairly similar to how we work other problems. But at this point, there's one important difference. Before you give your answer, we're going to divide boys and girls. Okay, so let's think about girls first, since they're on the top line. So for now, we're going to think about girls, and we're going to completely ignore the boys. We're just going to pretend they don't exist. So if we completely ignore the boys, there's only two possibilities for the girls. So half of the girls are big H, X big H, X big H. The other half of the girls and we don't have to keep using red and blue at this point. The red and blue is mainly just so that we can follow things up there. Now notice we have to rewrite this one a little bit, because when we have something that's heterozygous, dominant, allele, and recessive, we always write the dominant one first. So instead of X little h, X big H, I'm going to write that as X big H, X little h. So for the girls, half of them are X big H, X big H. Half of them are X big H, X little h. Now, what are we going to get for the boys? For the boys, half of them are X big H Y, half of them are X little h y. And I mentioned you could use percentages, so you could use 50% or something if you would rather, but you do have to have some indication of, what part of the offspring get each genotype and each phenotype. So basically, when we're doing our answers for the genotypes and phenotypes, we're thinking of it like two separate problems. Girls are one problem, boys are one problem. So now let's think about the phenotypes for the girls. Well, if we look, this girl is X big H, X big H. This girl is X big H, little h. So they've all got a big H. So all the girls have normal clotting blood. Now, what can we say about the boys? Half of the boys have a big H, half of them have a little h. So if we look at the boys, half the boys have normal clotting blood, half of them have hemophilia. And this is why we divide boys from girls, because a lot of times when we have X-linked characteristics, we don't get the same results for boys and girls. So think about if you were going to a hospital. Most hospitals now have somebody called a genetic counselor on staff. And you could talk to this counselor and say, well, there's this disease that runs in my family and my husband's family, and we want to see if we have children. What are the odds they'll inherit this? If you think about talking to a genetic counselor, they could tell you that 1/4 of your children would have hemophilia. But it would be much more informative if they would tell you, well, all the girls are going to be normal, but half the boys will get hemophilia on average. So you have to divide by gender once we get to the point where we're figuring out the genotypes and phenotypes, and basically work two separate problems. So while we're thinking about the girls, we're only going to think about these two cells in our Punnett square. While we're thinking about the boys, we're only going to think about these two cells, and we're going to ignore the two that have the girls in them. So any questions about incomplete dominance, codominance like we worked up here, or an X-linked characteristic like we just have worked on now? Okay, if there are no questions, I'm going to stop sharing, and I will go ahead and let you leave. There are some problems on here. The sample problems that we just worked are at the beginning of that section, but those are not due till Monday. And before you even start on them, I would strongly recommend do the regular Mendelian genetics problems so that you just feel comfortable before you get kind of to the exceptions to Mendel's rules. Any questions before you leave? No? Then I will let you go for today.